Okay, so good morning everyone. Can everyone see the slides?
Yes, sir.
Okay. So let us start our class today. And before starting our class, we will briefly look into what we have been studying in the last class. So in the last class, we have looked in. We were going through in these classes, few of the classes. We have been going through the pathways of the ethanol production, and we were looking into the basics of the ethanol fermentation process. So we have seen that whenever we are going to design, or for example, for any fermentation process. So this is one of the example that we are taking for the ethanol fermentation. Given any, for example, given any production of any particular bioproduct, so we have to look into several things. Like we have to look into what is the particular microorganisms you are going to take. Either the microorganisms has to be genetically engineered. How we make sure that whatever the byproducts that are being produced, so that are decreased, and how we make sure that. So we have first of all know the biochemical pathways along which by which the cells takes up the carbon source or nitrogen source, right? And converts that into product. So what are the intermediate steps? What are the enzymes that are responsible? What are the genes that are responsible? So we have to know those things. Okay, and also we have to know what are the substrate, which substrate are suitable for the microorganisms for their growth. So another thing very important is like you have to have that growth of the microorganisms, as well as that means microorganisms will produce the particular product. Okay, so we have to make sure that all the carbon source that is responsible for the formation of the biomass and the carbon source that is responsible for the formation of the product should be there. And along with that, the other things like the amino acids, proteins, which are nothing but the sources of. Hello, hello. Am I audible to everyone?
Yes, sir. Yes, sir.
Is slides visible?
Yes, sir.
Yes, sir.
Okay. So. Uh, So we have looked into these things, and then we have started talking about what are the solvent evaporations, and then we have started looking into that what are the characteristics of, for example, ethanol fermentation. So we have looked in that the ethanol fermentation is nothing but it is an anaerobic process. So what is anaerobic process? That means the fermentation is taking place in the absence of the oxygen. Okay, so that is very much important that when we are carrying out the fermentation, we make sure that we maintain the anaerobic environment. So we found that generally, but we know that cells grow in the presence of the oxygen, and it utilizes the carbon source, metabolizes it into the energy, and uses that energy for the growth of the cells and so on. So to make sure that we have the production of, for example, the ethanol, we have to make sure that the anaerobic condition, that is whenever we have a bioreactor system, it has to be a closed reactor, and make sure that whatever the oxygen is actually absent, because the presence of oxygen will actually inhibit the production of the ethanol. And why that because we know if we have the carbon source, and if we have a oxygen environment, what will happen? Most of it will be produced into the carbon dioxide and the water. Okay, if because that oxygen will completely oxidize the carbon source. So in this case, what is happening, since the oxygen is not present, so the partial oxidation of the carbon source actually takes place. And when we have the partial oxidation, we have the products such as the ethanol or other organic acids, like the methanol or gluconic acids or so on, citric acid, acetic acid. These are actually produced. So these are generally, so these are the products because of the partial oxidation of this organic compound. So generally we have generally the carbon source is the glucose, right? So in this case, we have the C6H12O6. So here we do the partial oxidations to produce the ethanol, that is nothing but the C2H5OH, right? So we have seen the overall reaction and yield of the ethanol production, and then we have seen that like there are different stains, particular microbial stains. So it can be some sort. So we have already seen in our earlier classes, what is the difference between the yeast, bacteria, fungi, and so on. So if you see the ethanol production, actually it takes place means both yeast, such as the Saccharomyces cerevisiae, as well as the bacteria, such as the Gymnomonas mobilis. Both of them can be actually used for the production of ethanol, and both of them have their own merits, demerits. We have looked into those things, and we have explained, like for example, means that both of them have the different biosynthesis pathway. Okay, Saccharomyces cerevisiae have a higher yield, the yield of to 90%. But in the case of the using the bacteria, the ethanol yield is 97%. Another thing to mention is that the formation of the, so ethanol is the product that is being produced. We have also seen that in the case of the ethanol fermentation, both the product inhibition is one of the issue. That means as the product is produced, okay, as a product means as the ethanol is produced, that ethanol at high concentration is going to inhibit the these particular microorganisms. Okay, so that is why it is the ethanol production is actually classified as a product inhibition. That means with the increase in the formation of the product, this production of the ethanol is inhibited. So what we do generally, so whenever the product is produced, that means the ethanol is produced, we take it out from that fermenter in a continuous process. Okay, and so we have seen in the case of the perfusion cultures or perfusion reactors, the product that is being produced is taken out, and in this case, for example, ethanol, so we do the distillation to remove the ethanol. So we heat up so that we can separate the ethanol vapors, and whatever the biomass is left behind is actually recirculated back into the reactor. Okay, so in that way, what we do is we make sure that fermentation doesn't this fermentation doesn't crosses the threshold concentration of the ethanol that is required, means that actually inhibits the microbial growth. Okay, so we make sure that in that bioreactor, that concentration of ethanol is not very high, by just continuously taking the ethanol out from the media and then supplementing back the what is the cells that is there, right? Similarly goes with the substrate. So generally, the substrate used maybe in this case the glucose. We make sure also that whatever the substrate is being added, that concentration is not very high enough that it inhibits the growth of that microorganisms. Okay, so another particular strain of bacteria or the microorganism is known as thermophilic species. So this is nothing but they have a high resistance to the temperature. Okay, and so as we have already discussed that in the fermentation process, when we are carrying out the metabolic activities, the cells are carrying out metabolic activities, there is a release of heat or the energy, and that actually increases the temperature of the growth media. So to make sure that we have, so for example, most of the bacteria grows or microorganism grows at suppose 35, somewhere around 30 to 37 degrees Celsius. Some of the, like most of the microorganisms have this optimum temperature for the growth. Okay, so in that case, to maintain that temperature, you have to have these heat exchangers that are installed along the periphery of these bioreactors, so which make sure that we have these type of temperatures. Okay, and so that adds to the cost and overall process. And if somehow there is some fluctuations of the temperature, so generally in the chemical, like this is one of the disadvantage of any fermentation process or microbial process, is that if you have a very high variation in the temperature, suppose +5 to -5 degrees Celsius, so this is actually going to, what will happen, it is going to kill the cells. Okay, and then that is going to affect the fermentation process significantly. Okay, so we have to make sure that the temperature, the pH, and all those things conditions that are required for the growth of the microorganisms are actually maintained in a means in a very accurate way. Okay, so that means that there is no effect on the growth of these microorganisms. Then we have looked into the substrate, different substrates we have talked about, starting from the lignocellulosic substrates, like the agricultural products, and each and every agricultural products have different types of the sugars, maybe a cellulose, sucrose, starch, lactose, and how they can be actually converted into the monomeric sugars, because as we already discussed, so these are nothing but disaccharides, or they are the polysaccharides. That means disaccharides means they are made up of two sugar molecules, and polysaccharides means it is made up of a long chain of the sugar molecules. Okay, so like in the case of cellulose, it's made of a long chain of the glucose units. So bacteria cannot consume this disaccharide polysaccharide. It needs to be in the form of a monosaccharides. Okay, so so that means so generally, if you are using different agricultural products or any lignocellulosic sources, it has to be converted back into some monomeric sugars like the glucose, fructose. Okay, and then only it can be consumed by the microorganisms, and will be used. Sorry, will be used for their metabolic activities. And so each and every, for example, this particular product, suppose the fructose 1 phosphate. So glucose, we know that glucose will, for example, if you look into the biochemical pathway, so glucose generally comes into this particular. So generally glucose converts into the glucose 6 phosphate. Now if you want to use the fructose, okay, so the fructose will enter into this pathway. So it has to enter through the fructose 6 phosphate. So generally what is done when we have the fructose is converted into the fructose 1 phosphate by a particular enzyme in the cellular machinery, and then fructose 1 phosphate is converted into the fructose 6 phosphate, and that then it enters it enters that biochemical pathway, for example, for the production of the ethanol. Okay, so like this, generally different sugars. So if it is a galactose, similarly if it is, so generally like cellobiose is converted into glucose, and this starch is converted into glucose, and sucrose is converted into the glucose and the fructose. So fructose enters at this point of fructose 6 phosphate, as we have seen in that biochemical pathway, and glucose enters so glucose gets converted to glucose 1 phosphate, and then it enters into the glycolysis pathway. Okay, so there are different means the different sugars are produced at different components are produced, and they enter at the different stages of the biochemical pathway. Now coming to the other sugars that we have seen, like the lactose consists of the glucose and the galactose. Okay, so means this glucose derivative enters the biochemical pathway, and then galactose is then converted into the in the pathways into the different components which enter into the biochemical pathway. So this process of breaking down uh, these disaccharides or the polysaccharides into some of the fermentable sugars is basically what was called as a catabolic primary metabolism, right? So we look into those things, and then we have looked into, like, if you are given a substrate, like the suppose you are given the rice husk or the sugarcane straw for the production of, suppose, ethanol. That ethanol now you are going to use as a biofuel in your automobiles. So the first step that we have seen is that the conversion of that substrate into fermentable sugars, and there are different steps that we have seen, that we need to grind, liquefy, or to carry out the saccharifications using the enzyme. And we have seen that the saccharifications is carried out with the enzyme called as the glucoamylase. So as the amylase name suggests, that it helps, amylase are generally is used for the breakdown of the starch. So generally those type of biomass having higher content of the starch, and so we use the one enzyme that called alpha amylase. So this alpha amylase actually leads to the production of the oligosaccharides. So oligosaccharides means the long starch chain has been broken down into the into, for example, some units of, for example, which is having 10 10 glucose molecules in the chain. Okay, then but again, as we told that oligosaccharides cannot be taken up by the microorganisms because it is still a chain of 10 molecules of, for example, the glucose. To break down further these oligosaccharides, so what we do, we add the glucoamylase, which breaks down these oligosaccharides to the individual glucose units. Okay, and this process of breaking down of the starch into the glucose molecule is called as the saccharification, and this sugar is then converted by the Saccharomyces cerevisiae, these glucose units, for the production of the ethanol. Okay, generally, I means whatever after the suppose the production of the ethanol using this agricultural biomass, we have some sort of a unfermented product that is present, and what is being done, so this is nothing but they're generally dried and they have been found to contain a high protein content, and this is actually what is called known as a dried distillers grains with solubles. Okay, so after the, so what is happening in this, so what is the main components present in this? So main components that are present are the unhydrolyzed cellulose. So generally the cellulose which has been, or for example the starch which has been converted into the sugars, that has already been taken up during the fermentation, but what is the left behind is the unreacted biomass that is left. Okay, so that is what is called as a dried distillers grain with solubles. Okay, and they're generally used as a animal fat. So then we have seen that, okay, the agricultural biomass is one of the substrate, but another important biomass coming up, which is though in the research stage, is to use the synthesis gases, like the gases which is being released directly from the industries, uh, which mainly composed of the carbon monoxide, hydrogen, carbon dioxide, or the ammonia, sulfur, hydrogen sulfide. So, what if we can use those emissions which are known to cause the global warming? If we can use those emissions directly for the conversion of the ethanol, and it was found to be very much successful. And some of the microorganisms like Clostridium, the orthoethanogenum, right, the Clostridium carboxydivorans, Clostridium Lujendali, and Clostridium rhizome. These are the few of the microorganisms which have been found that they can directly take the carbon that is present in the gases, such as in the carbon dioxide or the carbon monoxide, and then convert this carbon monoxide dioxide into the C2H5OH. So here in the ethanol we have the two carbons. So it combines, it utilizes the carbon from these gases and converts in them into the ethanol. One of the main challenges, so if it is possible, then why not this technology is actually commercialized at a large scale? But one of the main challenges that is there is the productivity, the percentage yield of this ethanol. So this is now significantly low, and the research is taking place to produce the microbial stains which can efficiently uptake these gases. So one of the challenge using the gases is the solubility of the gases. So if you see, a general fundamentally, you know that the solubility of gases are very low in the liquid media. So one of the challenge is to dissolve the means how much a gas will be dissolved, because if you see what is happening in a bioreactor, means these gases cannot be taken up by these microorganisms directly. Okay, so these gases first of all has to dissolve into the growth media. Okay, and after it is dissolved in the growth media, that growth media will mean that then only the cells will uptake the components, the dissolved components or dissolved gaseous components in the media by through the cells. Okay, so first of all, first step is the dissolving of the gases into the growth media, and the second step comes there. Now the dissolved gases that is there in the media will be taken up by the cells. So there are generally two steps how the carbon reaches into the surface or into the metabolic pathway of these microorganisms, and then once the cells have uptaken this dissolved these carbon components, then it enters into the cellular machinery of the microbes and converts this CO into the ethanol. Okay, so you can see that there are two resistances basically, and because of that, the first bigger resistance is the lower solubility, is the low solubility of these gases. So if there is a low carbon source available, then obviously that carbon source being converted into the ethanol into will be significantly lower. Okay. So this is one of the challenge why it has been it's still challenging in the part of the research to obtain the high productivity of the ethanol. However, different actually means different modern advancements is actually carried out in terms of the strain development. Okay, one approach is to develop the strain which can actually for example means easily that resistance that is there for the uptake of these gaseous components is can be reduced. Okay, by tuning for example the cell membranes of these microorganisms. And so another thing is so what will happen, another thing is the improving the solubility of these gases, and that can be done by incorporating some of the components within the growth media by optimizing the media and making sure that we have higher solubility of this carbon monoxide. We can improve the solubility of the carbon dioxide, carbon monoxide, or these carbon components into those media. Now coming to the biosynthetic pathway, we have already looked into the pathway, and we have seen there are generally two pathways that are being followed. One is the EMB pathway, which is known as embedded Meyerhof pathway, or it is sometime, or it is also called as the glycolysis pathway. Okay, so there are, we have seen, we will not go into these details today. We will go to the next topic, but we can see there is a cascade of biochemical reactions which is being catalyzed by particular enzymes, right? And there is a sequence of events that is takes place. So the main event is the conversion of the glucose into the pyruvate. Okay, and then the pyruvate actually will be converted to the ethanol, which actually takes place under the anaerobic condition. Okay, so under the anaerobic conditions, generally the means the pyruvic acid. So this pyruvic acid that is produced into the glucose pathway will enter these anaerobic metabolisms, in which it will converted to the this pyruvic acid will be converted to the acetaldehyde in the presence of the enzyme known as pyruvate decarboxylase, and this acetaldehyde will be converted into the ethanol in the presence of the enzyme alcohol dehydrogenase. Okay, so this is the route by which the ethanol production takes place, and if you see the overall rate of production, the 6 carbon glucose will be converted into the this 2 carbon ethanol. So generally there are 2 moles of ethanol that is produced. So we have here the 4 carbons, and 2 carbon will be 2 moles of the CO2 generated, which constitutes the 2 carbon. So generally the overall carbon actually remains balanced. So we have here 6 carbons starting from the glucose molecule, which is having 6 carbon, and then 4 carbons goes into the ethanol, and 2 carbons leads to the formation of the carbon dioxide. So overall there is a balance of the carbon balance is there. So so this is the overall means the biosynthetic chemical reactions for the production of the ethanol under the anaerobic condition. Now coming to the point, for example, in the case of the Xanthomonas mobilis, which is nothing but a prokaryote, it uses a different pathway, which is called as the KDPG pathway, which is nothing but 2 keto 3 deoxy 6 phosphogluconate pathway. Okay, so there is a slight modifications in this. So in the, if you compare the two pathways, okay, so if we compare here the two pathways, we can see the glucose in this case is converted into the glucose 6 phosphate, then fructose 6 phosphate, fructose 1,6 biphosphate, and then it goes into the glycolysis chains to produce the pyruvate. But in the case of the ED pathway, there is a slight change. And what is the slight change is that the glucose 6 phosphate is converted into the gluconate derivative of 6 phosphate, and that is going converted into the KDPG, that is the 2 keto 3 deoxy 6 phosphogluconate. And so this is being carried out by the so this pathway generally is being followed, for example, mostly by the yeast, Okay, but this follow pathway is generally mostly followed by the bacteria such as the Xenomonas mobilis. Okay, and so in this case, the main part of the main difference is thus the gluconate, the production of the gluconate, and why this gluconate production takes place. So if you see, if we start with the glucose molecule, okay, so here we convert the glucose 6-phosphate, fructose 6-phosphate, and so on. But sometimes these glucose molecules, in the presence of suppose that is suppose we have a very oxygenated environment, for example. So these in this case, these glucose molecules are converted into the gluconic acid. Okay, and that gluconic, if there is a high concentration of the gluconic acids in the media, for example, that the gluconic acid will be converted into the gluconate and the gluconate 6-phosphate. So generally in that time also we go for the we have this type of ED pathways. Okay, so we have to look into what is the difference of the ED pathways. So like how, so that is, and why the ED pathway, for example, is responsible. So we have seen in the earlier slide that Gymnomonas mobilis have around 97%, and the Saccharomyces cerevisiae, which is following this path, is having almost 90% yield of the ethanol. And we have looked into that why we have the higher productivity of in the case of the EMP pathway rather than the ED pathway, and that was because of the NAD plus and NADPH. So we have in this case the number of steps. So this is nothing but these are the compounds which are nothing but the electron acceptor, right? And that is something very much important in the for example the production of, or the biosynthesis pathway of for the production of the ethanol. So because we have anaerobic conditions, so generally there is a generation of these electrons, and as we are targeted towards the production of the organic compound, so this NADPH actually helps in the recycling of those electrons that is generated during the metabolic pathway. And so there are we have seen here instances 1, 2, and the third instance. So in the case of so there are three paths, three steps where there is a utilization of the NAD plus or the NADPH and so on. So so there is a means the depending on the concentration of the or the size of the electron acceptor that is there. So that will actually going to affect the productivity of the ethanol. Now coming to the next part, which is like how, what are the types of the fermentation processes we have. So generally, there are two types. We have the batch process and the continuous process. In the batch process, as we have already discussed, that we introduce all the components means the growth media, the microorganism, the cells, all together in the fermentation media at once, and then we carry out the fermentations. And what will happen? There is will be the production of the ethanol or any particular product, right? And after the means we take out the product only when the fermentation have completely means the fermentation is completely stopped. Okay, and then that is why it is generally carried out for 4 to 10 days. Okay, and if you see the parts and components, so we have very basic agitators and the impellers that is there. Okay, that makes sure that you have a uniform mixing of the cells with the growth media components, and there is no diffusion limitations. But generally we have talked about that the product that is produced is a toxic to the cells, the ethanol basically. So generally if you see most of the fermentations of the ethanol, they are either being done with the fed batch process, or it is based on the perfusion reactors. So what is the modifications that is there? So generally a modified batch fermenter is being used, as I suggested, that it's modified in such a way, means the batch process is modified in such a way that it makes it suitable for the production though. So how it is being modified? So generally the first step that is being done is to make that the sugar concentration or the nutrient concentration should be fixed within the fermenter. Okay, means for example, like if suppose the glucose concentration is something around 1 gram per liter is a threshold concentration, and product concentration, for example, something 0.5 gram per liter. So we make sure that a particular concentration is I mean that constant concentration is maintained by continuously adding the growth media component as well as continuously removing the product. Okay, so that there is no inhibition from the glucose end, and there is also no inhibition from the product end as the product is produced. So to carry out that, we need to continuously, we generally continuously separate or recover the products. Okay, so so in this case, generally the continuous process of the fermentation takes place. Okay, so, so like we have seen that the rate of sugar addition has to be regulated so that inhibitory levels of ethanol do not occur and cause the decrease in growth rate. So, this step is very much important. That what rate should the both substrate and the product should be added. Now, what are the substrate you have seen? So, what is that role of each substrate is something very much important. Okay, so if you see the carbon source, it can be the molasses and the starch. Okay, so we already discussed how the starch can be converted into some sort of a fermentable sugars, but there are some sort of a nitrogen source which are important for the ethanol fermentation. So generally they are like the corn strip liquor, soybean meal, pure ammonia or ammonium salts, urea, nitrate salt, and phosphate salt. So if you see from here, these are nothing but the chemical, when it's direct chemicals like the ammonia, ammonium salts, urea. Okay, but if you see the first two example, like we have the corn strip liquor and the soybean meal. So these are the very important points to highlight. Okay, so generally we have seen that for the carbon source we can go for the cellulosic biomass, but what about a nitrogen source? Can we take some nitrogen source from the environment, from the sources which are available, like the agricultural biomass or anything like that? So, or the industrial waste, for example. Okay, so so generally nitrogen source, for example, the soybean meal, means the soybean meal generally have a very high means the protein content, okay, and the amino acid content, which actually is the source of the nitrogen. Okay, and similarly goes with the corn strip liquor. So generally, this type of composition, what is the advantage and disadvantage? The advantage is that it consists of the mixed nitrogen source or mixed proteins or the amino acids. Okay, that is advantage because all the nitrogen source that are required can be supplemented by this type of corn strip liquor or the soybean meal. But the disadvantage on the other hand is the composition. So means generally when we talk of the word composition, we have to find what are the components present, right, as well as we have to find what is the concentration of that particular component that is there, right. So this is so in this case the conceivable soybean meal, it is very difficult to find the exact composition, and the uniformity of that composition is also very difficult to maintain, because depending upon the location, depending on the process, depending on the industries, depending upon the overall system, the concept liquor quality and the composition is going to be vary. Then there are other growth factors that are added during the ethanol fermentation. So these are very much important, some sort of vitamins or the micro elements like we have already discussed, that are or some of the cofactors so that are required for the that are required for the means carrying out of the growth, as well as that some factors required to carry out some of the steps in the biosynthetic pathway. So in the we have seen the biosynthetic pathway is a cascade of reactions. So uh, it may happen like some of the enzymes that are there may require some sort of a metallic cofactors, okay, or it may require some sort of vitamins for their activity. So generally these type of growth factors are being added, and the others are the minerals, like we have talked about the macro and the micronutrients. Okay, so they are required at trace quantity. Generally they are required a very less quantity, and that should be added specifically because of, as we talked about, that they may take part or play a role to maintain the enzyme activities that are for the enzymes which are responsible for the conversion of this glucose to the ethanol in the glycolysis pathway. Now there are different parameters that are known to affect the ethanol fermentation. So first on the parameter is called the temperature. So how does the temperature and what is the optimum ideal temperature that is there? Now we have discussed that we will be talking of the ethanol fermentation. Generally we'll talk about one of these the fuel, and the other is the for the for example the beverages, like we have the wine and beer. So generally the temperature, if we are going for the wine and beer productions, this has to be in the 20 degrees Celsius range, and we will look into this in more detail, like how this temperature is actually going to affect the quality, and also what is the means the different types of the products by just simply varying the temperature at which the fermentation is carried out. Different types of these beverages are can be produced. So we'll look into this in our next class. But if you see the ethanol production in terms of the fuel, for if you are going to use the ethanol for the fuel applications, so where the main objective is to produce the high yield of the ethanol with a higher concentration. Okay, but in the case of the, for example, the beverage, the obviously the aim is not to go with the very purest quality, but it is with some of the purest with a good product, which also consists of the mixture of other components. Okay, so the approaches for both application of the ethanol as a fuel and application of ethanol for the beverages, the fermentation approaches, though the fermentation remains the same, but the fermentation approaches are completely different. So we'll look into that. So generally, if it is for the beverage production, where the optimum is the 20 degrees. However, for the fuel production, it is generally at in the range of around 40 degrees, 40 degrees Celsius. Okay, so around 40 degrees Celsius is the ideal for that growth. So then generally some of the thermophilic modified thermophilic stains of the bacteria are used. Okay, then comes the term called pH. So we know that pH is very much important for the cellular growth. And if you see in the case of the yeasts, okay. So the means yeast, for example, because in the case of the yeast, what is going to happen is you have to grow the yeast will grow, and yeast generally grows in form of the molds. Okay, so initially, and also one other thing is the pH is something which is monitored in real time. Okay, and the pH has a significant effect in the cellular growth, their metabolism, and as well as the product that is going to produce. So generally the pH is actually monitored in such a way that depending upon the targeted production of the chemicals or the biochemical as such. Okay, so initially the media is maintained at a pH which is below 4.5. Although this pH actually is not optimal for the growth or the ethanol production. Okay, so now why the initial pH is maintained at a lower condition? Means it is because of the different media components that are present. Okay, sometimes the pH has to be maintained, or the optimum pH is calculated based on the media components that are there, because sometimes we do have some media components which is acidic in nature, or sometimes which is basic in nature. Okay, and if we, for example, suppose we have a citric acid or some sort of a lactic acid as a media component, and if you have them, so generally the pH will be lower. Okay, in this. So generally, if you see in the case of ethanol formation, the initial pH is at around 4.5. But if you see the what is the capacity of these particular yeast cells to grow and produce the ethanol, that pH range is from 3 to 8. So this is an optimum range at which the microbes can grow. Okay, at which the yeast cell can grow and produce, for example, the ethanol. And it has been found that by changing the pH, the amount of the organic acid products that actually being produced is going to, for example, change. Okay, so when we talk about the optimal pH, like when you are going to find out the optimal concentration of the pH or fermentation process. So it should be doing. I mean, it should be. It is. Means it is done by looking into the ethanol. Where we make sure that we have the higher ethanol production. We make sure that we have the higher biomass production or the biomass growth, and also by looking into the what is the physiological effects based on the waste products that are being produced. So whenever we are taking into consideration, so because sometimes the waste products that are produced, okay, that actually changes the acid. So for example, as I talked about that, sometimes the glucose is oxidized to the gluconic acids, like in the last example you have taken. So if it is converted to gluconic acid, so what is going to happen? That acid is going to decrease the pH of the medium. Okay, and so we make sure that the pH doesn't falls below a threshold concentration, which after which it actually going to affect the microbial growth of the cells. Okay, so that's why we have to always monitor the pH as a real time parameter during a fermentation, because the initial fermentation pH will be different because of the media components that are there. Okay, and then when the as we go along and carry out the fermentation, what will happen? That media components will be consumed up. Okay, and that media component will consume it, and it will release some sort of a proteins or for some sort of a byproducts or your particular product, for example. And that is, if suppose some byproducts or even the product is like if the product is a citric acid or a lactic acid. Okay, so it is obvious that with the higher productivity of these organic acids, what is going to happen? The pH of that overall fermentation is actually going to decrease, right? So this is a very important parameter, the pH, and at real time it has to be monitored so that it doesn't means crosses the optimal pH value that is required for the growth or the metabolic activity of the targeted microorganism. Now coming to the oxygen, if you see that the fermentation of this is of the ethanol is actually known as the facultative aerobes. So that means they grow well in the presence of oxygen, and when the oxygen is insufficient for oxidative metabolism, it actually undergoes the fermentation process. So what does it mean? So we already talked about like in today's start class that we need the oxygen, and the oxygen in the presence of the carbon will metabolize it to carbon dioxide and the water, and it releases energy which will be used for the growth of the microorganism. So if you want to increase the biomass concentration, suppose the x, okay, with respect to the time, we have to make sure make sure that the biomass is present, and that the oxygen is present, and that oxygen will be taken up by the microorganisms to increase the overall cell concentration and the cell counts. Okay, now if you are, for example, once you have an optimum biomass concentration with you, next step that we do is means we carry out the fermentation process. So what is the fermentation process? In this case, what we do, we maintain an anaerobic condition, especially in the case of the ethanol production. We maintain an anaerobic conditions, and under this anaerobic condition, whatever the cells or cell biomass that is being produced, it starts to it starts to follow that pathway from the pyruvic acid to the ethanol production, and starts for the initiates the production of the ethanol, okay, under the anaerobic condition. So it is what's very much, so both the things has to be minutely tuned, that we should have a high biomass concentration, and then that high biomass concentration is working on some sort of a carbon source in the absence of oxygen or under anaerobic condition, and produces the ethanol. Okay, so it is so very much important so that for ethanol production, the oxygen must be restricted from entering the fermenter, and zero to very low levels of oxygen in the broth should be maintained. So one of the thing, as we have discussed, that when we add when we have a sparger, for example, on the bottom, okay, and we passing, for example, the air or anything, so what is happening, that some amount of the oxygen will get dissolved into the fermentation media, some fraction of that oxygen. Okay, and that fraction of the oxygen may is also have been shown to inhibit the production of the ethanol. right? So to make sure that I mean, it is very important that we make sure that there is zero levels of oxygen in the broth. Okay, and how do we do that? So we do that by, so before we start our fermentation, along the spurger we try to pass some of the inert gases, like the nitrogen or the argon. What it does, it displaces the oxygen what is dissolved in the in the fermentation media. It displaces that oxygen, the dissolved oxygen, okay, and is replaced by the nitrogen and the argon molecules. And we know that the nitrogen and the argon means that actually helps to maintain the anaerobic condition. Coming to the ethanol concentration, we it is very much as we have discussed that what we have in this case, the product inhibition, right? So that means whatever the ethanol is being produced, that is actually inhibit the growth of the microorganisms, and the overall for example, the inhibition will be there in the microbial growth as well as the production of the product. So it is very important that we maintain the ethanol concentration, and generally it has been found that that a very small, like 4 to 6% ethanol actually inhibits the 40 to 50% of the of the yeast. Okay, and if you have a ethanol tolerant stain of the yeast, then the maximum concentration it can it can withstand is around 11 to 14% of the ethanol. Okay, so it is very much important that whenever the product is being produced, the ethanol is being produced, it has to be replenished, and it has to be taken out from that reactor. Okay, so that it doesn't overshoots this value. Okay, once it overshoots this value, what is going to happen? It is going to kill these yeasts, and the productivity will going to decrease. Like for example, in the case like 4 to 6% of ethanol actually inhibits the 40 to 50% of the yeast. So this, if 50% of the cells actually dies, this is going to severely affect, for example, the fermentation process and the productivity as such. Now coming to the fermentable sugar, so we have already talked that uh, a very high sugar concentration, suppose the glucose, is actually going to have a have a inhibitory effect or a negative effect, right? And this is because that of because of the osmotic pressure generally. So I am sure everyone knows like what is the osmotic pressure, how. So generally the cells always tries to make an osmotic balance. Okay, and if very high amount of the sugar is present, that high amount of sugar will create an osmotic imbalance, and sometimes that because of the imbalance, what happens, so the so generally cells have, if you see any microbial cells, they have a outer membrane, right? And those outer membrane allows the diffusion of the glucose from the growth media to the inside periphery of the cells, where this glucose is metabolized. Okay, so in this case, if we have a very high concentration of the sugar across the surface of this membrane, what it does is the cells undergoes an osmotic imbalance. Okay, and the cellular machineries, especially on the cell walls, which allows the diffusion of this glucose across this membrane, is actually restricted. Okay, and if and that actually leads to the inhibition of the carbon source, and that finally leads to the death of these cells of the microorganisms. So so it is very much important that osmotic balance is maintained, and this concentration of sugar that actually inhibits the metabolic activity of the cell is different for the different types of the cell, and it can be improved by carrying out some several modifications of the cells, by engineering the cells through some genetic engineering approaches or mutations, or by using the thermophilic the genes for the thermophilic bacteria to improve means not only the means to improve, for example, not only the thermophilic bacteria to improve the temperature at which it is working, but also the bacteria which can withstand the, or which can withstand the higher sugar concentrations but cannot convert that sugar into the ethanol. Okay, so use that to use those particular strains and carry out a genetic modification is something which is can be done to overcome this particular issue. And then comes the other requirements. Some of the other requirements like it requires one of the main thing is that there should be the balanced nutrient. That means that both the macro component of the nutrient as well as the micro component of the nutrient should be present, and that is something which makes it an ideal media for the fermentation. So generally some of the trace elements such as the zinc, copper, manganese, which are nothing but they act as some sort of a cofactor in the biochemical pathways like we have seen. And in the case, for example, for the ethanol fermentation, if you are working with the Saccharomyces cerevisiae, which is nothing but the yeast, it requires a vitamin. It requires a component called the biotin. Okay, and so it's nothing but a vitamin which is required for one of the biochemical pathway, which is nothing but the carboxylation reactions to stimulate the rate of fermentation and oxidation of the carbohydrate. So so this biotin actually helps in the carboxylation reactions. So generally we have the carbohydrates, right, the glucose molecule. So where we have to carry out the carboxylation modification at that site to make sure that takes place, the the biotin is required by the yeast. So this comes to that what are the factors for the ethanol fermentation. Okay, and this comes to the summary part of like how we can carry out. Means we have looked into, for example, uh, the substrate, the strain, the processes. Okay, how we have carried out those things, and what are the factors which actually improve by which we can select and improve the ethanol fermentation. Okay, so we will discuss this until this point for the today's class, and in the next class we will be continuing with the reactor systems that are required for the ethanol productions. Okay, so anyone any questions or doubt? Anyone?